Hello, everybody, and welcome back to another exciting episode about animal physiology. Today we're talking about neurons. So when you look at the animal kingdom, we start the animal kingdom usually with the sponges or the peripherans. Sponges don't move around much. They're not particularly interesting creatures in that regard. They tend to stay in the bottom of the ocean attached to something. But the next level up from there are the cnidarians, which include things like jellyfish, anemones, things that are a little more mobile, whether they're floating through the water or just interacting with other things. And that's where you start to see the beginnings of the nervous system. And specifically, they have something called a nerve net, just a sort of connection of various neurons together. And it's a very primitive nervous system. Nothing's been centralized yet. So the big question is, why evolve something like a nervous system in the first place? And how do you think it evolved? What are the conditions that allowed it to start and to initiate? What's the point of having a nervous system? And how did the environment influence that? Well, the more astute among you might have picked up on the fact that movement is a big part of it. The general consensus among scientists is that the nervous system basically started as a way of controlling and coordinating movements across different parts of the organism. You also, in the process of doing this, start to develop new types of cells. A sponge is a very simple creature. It has, it's almost a colonial microorganism in that you have all the cells, they're all pretty similar, doing similar jobs, and depending on where they're located, they'll do that job. But when you start getting up into jellyfish and anemones, you start seeing specialized tissues, cells doing particular things. But they still have to be coordinated and integrated among each other in terms of being able to move the entire creature as a whole. So it makes sense that you would evolve some sort of way to do that. And the nervous system is a very, very clever way to do something like this. And then on top of that, if you're moving through your environment or you're interacting with your environment, you run the risk of having to uncover new stimuli. So you need to be able to perceive new stimuli and understand them. And thus the beginnings of what are going to become the two big branches of the nervous system, the sensory component and the motor moving component, the locomotion component. So let's start first with a quick little question. Here's a neuron, sort of your classic neuron, and here are a bunch of letters indicating particular structures and a bunch of numbers. So take a second to see if you can figure these things out. I'll give you basically a second. All right. Hopefully you've been able to figure it out. You've probably gotten this at some point or another. So let's just start with the number one. Number one obviously is pointing near the nucleus there. That's going to be the cell body, or soma as it's known in the nervous system. Right, so that's the soma, so that'd be D right there. And then right by it, you have number two. Number two is where the soma starts to taper off into a long projection, and that coincides with the hillock, or the axon hillock, E. Then number three, you have this long projection, which we already said the axon hillock, so that's the actual axon itself, right? So that would be B. 4, that's where the axon ends, what we call the axon terminals, or C. And then 5, you're looking at little spots on the axon that are exposed, and those represent what's known as the nodes of Ranvier. And the nodes of Ranvier are spots that, well, we'll talk a little bit more about them in a bit, but they're very important to signaling speed and things like that. And then 6 points to these other cells, these yellow cells that surround the axon, and these are glial cells. These are sort of accessory cells that are also found in the nervous system. Your nervous system basically has neurons and glial cells, and glial cells do a whole bunch of different things and have a whole bunch of different morphologies. Very unique. And then finally, number 7, well, this is where the whole process begins. Number 7 represents the dendrites, or A. And this is where the input comes in. So neurons have an input section, a conduction section, and an output section. And this is very important because this means that neuronal signaling is happening in a unidirectional fashion. Input comes in one way through the dendrites. The decision as to whether or not the signal gets carried is made at the level of the soma, or the axon hillock, in what we call the integration zone. It gets sent through the axon, and then eventually at the axon terminals, it then gets sent out as output to stimulate the next neuron. And you probably know that neurons work by working together, forming synapses, and there's a lot of different ways, as this picture shows here on the right, that you can set up synapses. It really doesn't matter how they're set up. It might affect speed or the ability to take in multiple signals. But in the end of the day, the signal is being sent in a unidirectional fashion, so in order to develop any sort of complex response, you need to develop a circuit. So by interacting these things in particular components, you're going to get a signal being sent. So in the end of the day, the neuron is operating like an electrical conductor, right? And your nervous system is just an electrical system. And you know that with electricity, you can do anything, right? You can create a small little device. I like to use the example of a hairdryer. A hairdryer is pretty straightforward. It has one thing that it does. It warms up hair and produces air. And that requires a little bit of wiring to be able to do that. If you have more wires and develop more circuits, you could have more complex functions, like say in a tablet or your smartphone, which can do a lot more stuff because it has a lot more circuitry involved, right? So the complexity is going to come from how complex the circuitry of neurons are operating, right? So it's their environment. So we're talking about an electrical system, but at the same time, across neurons, you have to send the signal through some kind of chemistry, right? So the chemical synapses, electrical conduction. So it's pretty ingenious the way this works. So how did we figure this out? The collective we, of course. So Camillo Golgi, famed for Golgi apparatus, he developed a method to stain nervous system cells as compared to glial cells using silver nitrate. And he noticed the interconnectedness of these cells. And in doing so, he reasoned that there must be some sort of net. Now, we've already talked about the nerve net, so it wasn't that far off. This idea that your central nervous system and your nervous system in general is operating through just a net that kind of, can activate anywhere, right? And he referred to this as the reticular theory. So his big contribution is that he found that there were connections, and there were a lot of them. Now, this was challenged by Santiago Ramón y Cajal, who said that, there isn't this continuity of cells that just kind of like energize the whole system, so to speak. But in fact, you have cells that are specialized and they interact with each other in particular ways. So he moved the emphasis from this big net that kind of covers everything to having a more neuron-based approach, right? So what he termed a neuron doctrine. And turns out he was correct. But because both of them made a contribution, they famously both earned the Nobel Prize. And Golgi was very adamant about his particular theory. He was particularly trying to basically start a beef with Ramón y Cajal. Didn't get anywhere, but they famously kind of had to accept the award together, even though they didn't like each other. It happens, right? People can get too competitive sometimes. But nonetheless, the neuron doctrine is what we accept today. So when we talk about neurons, we like to usually draw something like a motor neuron or a multipolar neuron. This is the type of neuron that's going to send signals away from the central nervous system through the peripheral nervous system to cause some sort of effect, right? And this could be an effect of causing a muscle to contract, but it could also mean something like causing a gland to secrete something, or causing, some sort of constriction somewhere, or some kind of release somewhere. Who knows? So signals that are carried away from the central nervous system, we use the term efferent to describe those type of signals, as opposed to afferent signals, with an A, that are bringing information to the central nervous system. So when we talk about that, we're usually talking about the sensory component of the nervous system, right? So in a sensory neuron, looks a little bit different because it doesn't have the cell body right in the middle of the thing. It actually wants to send that signal as fast as possible. So it decreases resistance by moving that cell body out of the way. So the conduction zone is much quicker, right? The conduction happens at a lot quicker velocity than an afferent signal. So again, afferent, bringing information to the central nervous system, which by the way you should know is the brain and the spinal cord, and then efferent is taking it away from there. Now, what happens when it's in the central nervous system? Well, it's interacting with interneurons. So interneurons are basically what the central nervous system is made of. These are neurons that are going to connect, and their main job is to connect with each other and to provide some level of complexity in terms of circuitry, right? So often these neurons are going to be bipolar. They're getting signals from both ends, right? So they're getting information and it's being sent across all these different ways, all these different pathways, and they're all interconnected. And the more interconnectivity you have, the more connections you have, the more complex a signal can be sent, a more complex thing can be produced, right? Something like a thought, for example. If you're really, really complex, if you look in the brain, especially in areas of the cortex, areas where there is often a lot of learning, and we'll talk a little bit about learning and what that really means, there you have often neurons that can have extensive branching. So here's an example of a pyramidal cell. These are the type of cells you might see in the cortex, type of cells you might see in the midbrain, in places like the hippocampus, for example, places that are getting information, right? So think of like a memory cell, for example. You're forming a new memory and you're making associations with that memory all the time. Sometimes particular scents or sounds can trigger a memory. All that information is being coded through branching and circuitry. So a cell that encodes that memory and compartmentalizes that memory, you can retrieve it because it's got a lot of branches everywhere. And it's kind of got its fingers in a lot of pies, so to speak. Okay, so one thing about neurons is they have something called a resting membrane potential. And a potential, in its simplest form, is basically a difference in charge across a membrane. So everybody knows cell membranes have, some ions outside, some ions inside at varying concentrations, and that sets up a difference in charge, right? So it might be more negative relative to the outside on the inside of a cell, vice versa. And that is what we call the potential. So think of it as the potential for ions to move back and forth. Now, you probably know some stuff about diffusion. You probably know something about the movement of solutes, or ions in this case, and the direction they're going to move. So the resting membrane potential of a neuron, we often quote it as being 70. Right, minus 70 millivolts, and that minus 70 millivolts simply refers to the fact that a reference electrode on the outside of a neuron is going to have about a 70% difference with what's on the inside in the recording electrode. So the difference between them is about 70 millivolts, right, with a slight more negativity on the inside than on the outside. So the question is, what accounts for that? So here are 4 possibilities. Take a second to see if you can figure it out. Okay, well, long story short, you have sodium ions on the outside and you have sodium ions on the inside of a cell. The concentration of sodium ions is much, much greater on the outside than on the inside, right? So that membrane, there's a big difference in sodium concentration, more of it on the outside and on the inside. The opposite is true of potassium. Potassium, there's more potassium on the inside of a cell than there is in the extracellular fluid. But the problem is you have this constant leaking, potassium leak of these ions, potassium ions, and they're always leaking in a passive way. So there's this constant net loss of positivity, and that constant net loss of positivity is actually what sets up this difference in gradient, right? So you have a membrane that's impermeable to sodium, but is permeable to potassium. And potassium is going to follow its natural gradient. It's going to go from an area of high concentration inside the cell to an area of low concentration outside the cell. Well, that's not good, right? So the cell has to somehow maintain an equilibrium. So it does this by having a sodium-potassium pump. And the sodium-potassium pump works by basically taking sodium, exporting it out, and then bringing back potassium into the interior of the cell. And this is a process that requires potassium to be pumped against its concentration gradient, right? And because of that, you're going to have to invest some energy. So ATP is used for this. So this is the reason why energy is such an important commodity, is because your cells are constantly having to maintain this resting membrane potential. They have to maintain this gradient potential, electrochemical gradient potential across the cell membrane. So there's constantly investing ATP, ATP, ATP into doing this. And this is costly. And this is the reason why cells have to produce most of their ATP. Believe it or not, it's estimated that definitely more than half of the ATP investment is just simply in maintaining this resting membrane potential. Because if you don't maintain it, neurons can't fire, things can't happen, and yeah, long story short, nothing happens. There's no physiology, right? Neurons don't work, other cell functions don't work, there's no gradient, right? Because things are going to eventually leak out and it's going to equalize. So the correct answer in this case is going to be C. The passive outflow of potassium ions and the active pumping of ions back in creates the electrochemical gradient that is necessary. So, pretty long-winded answer, but very important. Resting membrane potential, shout out to all the ions out there. So, sometimes the resting membrane potential changes, and when it does, things happen. So neurons are famously known to fire, and what that simply means is they have an action potential that happens. In order for an action potential to happen, it has to start somewhere. So every time a neuron interacts with another neuron, right, at the synapse, what happens is it's getting a signal. The signal can be something that causes it to want to fire. It's excitatory, and what that means is that it depolarizes the cell. And what depolarization means is that the cell membrane becomes a little more permeable to sodium, and sodium rushes in because, again, there's more sodium on the outside in the extracellular fluid than in the interior of the cell. Or it could be inhibitory, in that it causes it to hyperpolarize or become more negative. So, again, depolarization, sodium's coming into a cell, so that cell becomes more positive. Now, because our starting point is already pretty negative, what that more positive means is less negative. So again, think of it on that scale. Hyperpolarization means it's becoming more negative. And the reason it's becoming more negative is because it enhances the loss of potassium, right? So it opens up more potassium channels, causing that stuff to leak out of the cell. Because again, there's more potassium inside the cell. So these are called postsynaptic potentials, or PSPs. These are the things that happen to a neuron when it's getting information from another neuron. Now, these can also build on each other, and as a result, we say that they're graded potentials. So when we talk about something being graded, we talk about it being graded on, a scale, right? So you can get a 70% on the test, you can get a 75% on the test, or an 80% on the test, right? Or 90% on the test. That's a continuous scale. If you have excitatory postsynaptic potentials, and then you get another one, and then you get another one, and then you get another one, they're going to build on each other. Eventually what will happen is you'll cross a line which will cause that action potential to happen. And it's an all or none phenomenon. A neuron is either going to fire, have an action potential, or it's not going to fire. And what determines that is whether or not it crosses some threshold. And depending on the type of neuron you have, that threshold can vary. Now, when this threshold does happen, or when the threshold does get crossed, that neuron's going to fire. And when that neuron fires, your action potential has just occurred. So this is an example showing across very short span of time. Again, we're not even talking seconds here, we're talking milliseconds. Changes in membrane potential. Membrane potential is the charge difference across a membrane, right? So we usually use V to abbreviate this, volts, right? So volts just means the potential for something to happen. So if you have a 9-volt battery, that means that there's a potential for 9 volts worth of current to move across something, right? It can power something with that much if you have enough of a battery to do so. So it's a measure of the potential, the difference in charge from one spot to another. In the case of a battery, like the type you would put into something, like a remote control or something, you have a positive end and a negative end. Well, the difference between them is 9 volts in a 9-volt battery. It's 3 volts in a 3-volt battery. It's 125 volts in a 125-volt battery, if that makes sense. So membranes are going to be permeable to ions at different rates, depending on a number of different factors. in particular the difference in concentrations across the membrane, right? So this is showing the trace for a particular neuron. So at time point 0, which is that first little red dot there, you're at the resting membrane potential. Nothing much is happening, right? You have a potassium leak, you have a sodium-potassium pump bringing that stuff back in. But then you start getting little disturbances. And as you get little disturbances, in this case, excitatory postsynaptic potentials, you see that blue line is getting a little closer to that red line. And that red line presumably is your threshold. So as you get closer to that red line, once you cross it, what's going to happen is all of a sudden, what started as a few sodium channels opening here and there, now all of a sudden a whole bunch of sodium channels open up in the cell. And then because of that really big gradient that's maintained in sodium across the membrane, you're going to get a flood of sodium into the cell. So that's what's causing it to now shoot from minus 50 millivolts past the 0 up into the plus 50 in this case. So sodium channels are opening on that first part of the curve. Then what's going to happen is enough's enough. You're getting close to some kind of sodium equilibrium. That big rush of sodium has happened. Those sodium channels are now going to close. That's an unstable condition. So the cell is going to want to react to this. And how it reacts is then by opening up potassium channels. So it went from being at a resting membrane potential, it depolarized, became more positive, right, by opening up those sodium channels. And then now those sodium channels have closed up at the apex of that curve, that blue line, right? I think it says 3 there. And then potassium channels are going to open up. So 4 is showing the repolarization, where potassium channels are opening up, and now potassium is going to leak out en masse out of the cell. And as it's losing those positive potassium ions, it starts to get back into the negative, right? So it gets back and actually dips below the threshold and below even the resting membrane potential. So by that point, what's happening is it's now at number 5, and at number 5, it's hyperpolarized because it's basically let out a lot of potassium and didn't close it on in time. That's the easiest way to think of it, right? So it overshot the mark. Then what's happening is it's basically going to start to recover back into its resting membrane potential because it's closed off its potassium pump, it's closed off its potassium channels, and now that sodium-potassium pump is working to restore back into a resting membrane potential. And then once it gets back there, the whole process could happen again. This happens very, very quickly. So again, resting membrane potential, sodium channels open, depolarization. Sodium channels close, potassium channels open, repolarization. Overshoots the mark, potassium channels close, but now you're hyperpolarized, and sodium-potassium pump kicks back to start restoring membrane potential. Pretty simple if you think about it. So the rate with which this is going to happen, again, I talked about the potential. Well, Ohm's law states that the voltage across 2 points, in this case with a membrane, is going to be a product of the current, right? So current is the ability to conduct an electric current between 2 spots. So the way to think of it in this case would be the potential is the concentration gradient across the membrane. The current represents the number of channels open, let's say. And then resistance, right? So resistance is the capacity for ions to move across the membrane, right? So V equals IR. V equals IR. That's Ohm's law, and that's a very big determinant of neurons and how they work. So people have tried to mathematically figure this out. And Nernst was the first. He came up with the Nernst equation. And the Nernst equation basically takes chemical concepts and applies Ohm's law to it. So it's focused on determining the equilibrium potential of an ion. And it works one ion at a time. So the equilibrium potential, the membrane potential of Bm, can be boiled down to these various constants. Now again, this is something you would figure out in the lab with a preparation of a neuron that you can then manipulate concentrations of things. But basically you have the equilibrium potential of X ion, right? Ex equals RT R is that gas constant, right? T is temperature in Kelvin. The F is the Faraday constant, which is 96,490 coulombs per mole, right? And this is a constant, so it doesn't change. And then depending on the type of ion you're working with, now in the case of sodium or potassium, we're talking about a +1 ion. If you're talking about chloride, it could be a -1. If you're talking about zinc or some other ion, it would be 2+, right? So you're looking at that and then times the natural log of the ion concentration on the outside over the ion concentration of the inside. So again, this is basically for one ion at a time. This whole process can be simplified, and if you were to simplify it and then convert it to a regular log, E, equilibrium potential, will equal 58 times the log of ion concentration outside over ion concentration inside. So here's a quick question. What's the equilibrium potential if the potassium ion concentration of the outside equals that on the inside? Well, this is where logic and reasoning work out over having to calculate it. So you could throw it into your calculator and you could just pick a concentration. You could say 1, you could say 5, and be equal on both sides. But either way, you would end up with 1. And the log of 1 is 0. So if the log of 1 is 0, the equilibrium potential is 0. But you don't need to actually do the math, because it makes sense. If you don't have an equilibrium electrochemical gradient, if the concentrations are the same on either side of a membrane and they're in equilibrium, then yeah, the equilibrium potential will be 0. There's no reason for those things to move. So that's Nernst equation, one ion at a time. But we know that nature doesn't work that way. Nature has multiple ions that are often working at the same time, and all of them contribute to the voltage of a neuron, because a neuron relies on multiple ions. So Goldman expanded this equation to include multiple ions. So he took it from a one equation thing to a multiple equation thing. And you can simplify it in the same way, 58 log concentrations. But in order to figure this out, they had to obviously verify it. So one creature that is particularly important in neuroscience and in electrophysiology in particular is squid. So squid have a very large axon, a nerve that runs down their mantle, and it actually is involved in contracting the fins that allow them to propel through, right? So they're called the squid giant axons. And they're giant because you can see them with the naked eye and you can manipulate them, and they can be dissected out very easily in this invertebrate, right? And it allowed them to do a lot of studies where they can basically, propagate signals and then measure things like conduction velocity. They can manipulate concentrations and do this, that, and the other. So this is an example where the squid giant axon, not to be confused with giant squid axon, which just means giant squids, they're a real thing, and they probably have enormous axons, but don't worry about them. Any old squid will do in this case. They can actually validate the equation by manipulating it in the lab. So they can change the concentrations inside and outside of the axons and actually see what the voltage changes are. Right? So this is a great example where a animal model validated a mathematical model. So collectively, we often refer to this as the Nernst-Goldman equation, just to give props to both. But the difference here is that you not only have the concentrations on the outside and on the inside of each individual ions, but you have to have some sort of relative measure of their permeability across the ion. So that P function in the equation represents permeability of that particular ion, which basically means how many channels are there for this thing, right? Is it something that can move a lot and easily because there's a lot of channels for it, or is it something that doesn't have a lot of channels and it's relatively impermeable across the membrane, right? So notice K plus outside, inside, Na plus outside and inside, the numerator and the denominator. But when you get to chloride in this case, when you look at chloride, the inside is the numerator and the outside concentration is the denominator. So whenever it's a negative ion, right? A negatively charged ion, you just switch those. But notice the P doesn't change, right? The P is going to be the same number inside or outside. It's just a measure of overall, can it cross the membrane, regardless of which direction it's going. So you can do this, you can figure it out, and actually validate it. And I'm sure we'll go over some questions doing this in class. Think of neurons as being all or none. They fire or they don't. Now, when a neuron is firing, it can't fire at, it's already firing, so it's not going to fire again. So we refer to that time when the sodium channels are open and the potassium channels are open, the depolarization part and the repolarization part, as being an absolutely cannot fire period, or what we call the refractory period, the absolute refractory period. So regardless of stimulation, you can't have that neuron fire again. It's all or none. Once it's firing, it's firing. Now, during the hyperpolarization phase, when it's trying to restore back to the resting membrane potential, it's a relatively refractory period, meaning that if you were to stimulate this neuron again with another stimulus, whatever, regardless of what that stimulus is, it could fire again if it can get over the threshold, which means that depending on the strength of the stimulus, it can or cannot fire. Now, normally it wouldn't fire, so we call this a refractory period. Refractory period just means the time in between. So until it gets back to that resting membrane potential, it won't fire. But if you have a big enough stimulus to get it over the threshold, it could possibly fire. It's just much harder. But it's not impossible. So the ability to restore normal often determines how fast a neuron can fire again, right? So fast-acting neurons can restore that normal quickly. So here's some data from 4 species of squid. The species names are shown there. You see the different symbols for them. And in this study, they looked at conduction velocity. How fast did the signal get sent down the axon? And they tested these conduction velocities at different temperatures. So they had this, the squid giant axon prep, and they basically measured conduction velocity, how fast can it send a signal across a certain span of time, and across different temperatures. So here are the 4 species. Notice one of them does not belong. All right, so you got 3 species of Loligo, that's the genus of that particular type of squid, and then you got the Sepioteuthis. Sepioteuthis sepoidea is a different type of squid, right? So it's related, but not as closely related as the other 3. So what do you notice? Well, looking at this data right off the bat, you can kind of see that if you look at the chart, you have conduction velocity against temperature. As temperature increases, conduction velocity increases. So right off the bat, the warmer it is, the faster the ability to send a signal. Makes sense, right? These are cells after all. But you might also notice that the 3 species that are similar, the 3 Loligos, kind of cluster together, whereas Sepioteuthis is out on its own, and it's a little slower, right? So regardless of temperature, it's always on the low end. So there's differences between Loligo and Sepioteuthis, and obviously 3 closely related species are going to be most similar in this regard because they evolved from the same ancestor more recently, so they'll share a lot more similarities, right? Just basic evolution for you. Sepioteuthis, even if it's a squid, will be a little bit different because it's just not as closely related. Diverged a lot earlier. But the big difference, and the reason accounting for this, is that Loligo has a lot more, is a lot larger species, and it has larger axons. And having a larger axon means you have a larger axon diameter, and a larger axon diameter means there's going to be less resistance, right? So why does axon diameter impact conduction speed? Well, the larger the diameter of your axon, the faster your conduction speed, because you're going to have decreased resistance. So just like when we talked about the circulatory system, and we talked about how in the circulatory system, the vasodilation, vasoconstriction will change resistance to flow, right? And if you're more vasoconstricted, i.e. you have a smaller diameter, you're going to have a greater amount of resistance, a contact with the outside, as opposed to the middle, where there is no contact with the outside of the wall. Whereas a larger one, you'll have more volume and less volume in contact with the surface, so there's lower resistance and you can go faster. The same thing applies to axons. So this is showing different diameters of axons and conduction velocities. So generally speaking, on the x-axis, the further you go to the right, the curves and the points tend to be a little bit higher. Makes sense. So larger diameter, lower resistance, faster conduction velocity. The other big thing that influences how fast conduction can happen down an axon is whether that axon is myelinated or not. Myelination is basically insulation of an axon. So when we talk about myelin, we talk about the myelin sheath. And the myelin sheath are those other glial cells that we talked about. So glial cells are basically any other type of cell found in the nervous system. Sometimes they're involved in immunity because the nervous system has its own immune system, because things from the blood, cells from the blood can't get into the nervous system. So it has its own type of glial cells that are involved in immunity. It has glial cells that are involved in nutrition, that provide metabolic products and help to nourish the neurons because they're very energy dependent. So they produce things like glucose and kind of give it up to the neurons, and then get imported into the neurons and used for metabolism. But when we tend to think about glial cells, we often think about their role at acting as insulation. So in the central nervous system, the CNS, you have oligodendrocytes. In the peripheral nervous system, you have other types of glial cells called Schwann cells, but they both work the same way. So in that image right there of that neuron, you see how the myelin acts. It basically just kind of wraps around the axon, and one axon can have a whole bunch of these glial cells wrapped around it. When it wraps around it, it's basically doing insulation, right? Just like you have a wire that's, you have the copper wires that run through, but then you have the plastic part on the outside that acts as insulation, because you don't want them touching each other, right? They'll short out. The same thing with the myelin. Now, whenever you wrap something and you insulate it, you increase the speed of conduction. And in the case of neurons, the way this works is you basically have a wave-like opening of sodium channels as a neuron fires. So the neuron fires, it starts at the axon hillock, sodium channels open up, and then the sodium channels all along the axon start to open up. And the whole thing just propagates through the axon, happens very quickly. And these local openings are, they refer to the Hodgkin cycle. The Hodgkin cycle is basically something where you are opening a channel, disturbing the electrochemical gradient in that area, and that causes other channels to open, and then open, and then open. So it's kind of like a wave. You go to a baseball game, and everyone's doing the wave. It starts in one spot, and then just waves down. Everyone just kind of like responds to their next door neighbor that's doing it, so they go ahead and do it too, right? And then the whole thing propagates through the stadium. The same exact thing is happening with ions in the axon. What the myelin does is it basically closes off a bunch of areas so those channels don't open. Instead, you have channels located in the nodes of Ranvier. Those are the exposed parts of the axon. So what happens is whatever sodium floods into the axon at that part where the sodium channels opened up, what's going to end up happening is those sodium ions are then going to just flow down the axon and then basically jump all these little spaces where the myelin is. And because it looks like it's jumping, they call it saltatory conduction. Because on the outside, it looks like you're basically just, jumping over the axons with the opening of the channels. But what's actually happening is, the thing's a tube, so sodium ions are moving down the tube and causing that activation. But it's happening a lot faster because you don't have to open up all the channels. It's basically, imagine like doing the wave. You have one stadium doing the wave the normal way, and then you have another stadium where one section does the wave, and then 2 sections over they start the wave, and then the other one 2 sections. So it skips a couple sections of the stadium each time. It's going to be faster. They'll get to the endpoint faster. That's basically all it's working. So this is showing some just some relative speeds, right? So a small unmyelinated axon could have a 2 meter per second, which is still pretty fast. But if it's myelinated, it can jump up to 15 meters per second. So it's a big jump in conduction velocity. At the same time, myelinated neurons, if they're also enlarged, can be 120 meters per second. So we're talking about big differences with the result of myelination. So very cool. So whenever myelin is lost, that often slows down conduction of axons. So, for example, multiple sclerosis, the sclerosis comes from the hardening of the myelin sheath. It becomes ineffective, and basically they're losing their myelinated axons, so certain processes get affected, right? And it's an unfortunate disease. My sister actually has MS, and just there'll be days where she can't do things, right? And it's because of this nervous system effect. But moving on, something a little more happy. Let's talk about synapses. Let's start with the type of synapses nobody ever talks about. These are ionotropic synapses. Ionotropic synapses, we've seen them before. So think of like heart muscle, for example. You have gap junctions. Gap junctions are basically complexes of proteins that are called connexins or innexins, depending on the type of creature you are. And long story short, they're kind of like what we just talked about with sodium channels. What they'll do is they're basically connections between neurons or muscles and ion transfers through these gap junctions, causing very rapid depolarizations. So this often produces very fast IPSPs or EPSPs, and they're very quick because they basically open up channels directly. So you can have a synapse where all of a sudden a depolarization is going to activate a bunch of channels to open up and cause this thing to happen, right? Again, this is basically what's already happening inside of a neuron, but this is now a synapse. So this is something happening between neurons. The depolarization and channels of one are affecting the channels of the other. No chemistry is involved, right? So in this case, these are very quick, which suggests that there's very low resistance because you're moving the ions directly from one to the other, right? So there's low resistance. The membrane's not even really involved at this point. So when we think about these type of synapses, we often think about 2 players. EPSPs that are fast, we often think about the central nervous system and glutamate. Glutamate is a neurotransmitter that often uses these type. We'll talk a little bit more about their receptors, but they'll basically activate these receptors, and they're the main stimulating neurotransmitter in the central nervous system. So you think about your brain function, your brain's firing, your neurons are firing in your brain, this, that, the other. A lot of that is actually glutamate. So the stimulating process, right? They're not always stimulating and excitatory. Sometimes they are inhibitory, but for the most part, we associate glutamate with being this excitatory neurotransmitter. And the way they excite is they basically increase sodium potassium channels, right? So they increase the sodium potassium influx, increase sodium potassium outflux, and they do it very quickly by directly opening these channels. On the flip side, right, so the yin and yang, you've got glutamate as the main excitatory, GABA, which is or glycine is also called, so it's gamma aminobutyric acid is what the GABA stands for, glycine aminobutyric acid, and it's the main inhibitory neurotransmitter. So your brain is always working, and sometimes it needs to calm down, and GABA is the main neurotransmitter that causes that. And the way it works is by activating chloride channels. So by opening up these channels, it causes chlorine ions to flood into a cell. And in doing so, makes it basically hyperpolarizes it and makes it less likely to fire. And this is important because when you have these sort of EPSPs and IPSPs, they're basically trying to restore a balance, maintain function. So kind of like in the heart, where your pacemaker is making your heart beat very, very quickly, right? And then you have your vagus nerve is causing it to slow down to maintain a regular heartbeat, right? But if you need your heart to beat faster, like say if you're stressed about something, then that inhibition will be lifted and it'll start to go faster. That's kind of how the brain works too, right? Glutamate is always stimulating stuff, but GABA is helping to slow it down and inhibit it. And often when you have a loss of, say, GABA receptors or GABA signaling, you'll see processes in the brain just happening a lot quicker, right? Or a little more spastic-y, right? I think of, for example, Parkinson's disease. Parkinson's disease, a characteristic of Parkinson's disease is kind of uncontrolled tremors and the loss of fine motor control. Well, that comes from GABA being lost over time, and it's causing, a lot of things being, motions that would otherwise be inhibited are now stimulated. So you have a loss of fine motor control, right? So depending on the ion, they all playing their part to help maintain balance in the nervous system. It's quite a wonderful thing. So that's synaptic transmission in a nutshell. So the signal is being sent down. So you see here in the thing it says 1 action potential reaches terminal. Yeah, it's going to do that. And then up until now, it's been sodium channels that have opened up. But now what you're going to get is the Hodgkin cycle is going to activate calcium channels. So in the axon terminal, you have a lot of calcium channels. And when these get activated, it basically causes a rush of calcium into the axon terminal. Calcium is very important because calcium actually binds to the area where the synaptic vesicle that's holding all the neurotransmitter inside the axon terminal is going to merge with the cell membrane. and cause the exocytosis of that neurotransmitter. So that's actually regulated by calcium binding. So calcium is necessary for this step. So that the synaptic potential, you have neurotransmitters being dumped, it fuses with the cell membrane. A lot of debate about whether or not it's a complete fusion or whether it's just a quick dumping and then it gets picked up again. They'll sometimes talk about the kiss and run model of calcium regulated exocytosis. Doesn't matter. The point is the neurotransmitter is going to be dumped into the synaptic cleft, and waiting for it on the other side of that little chasm is the receptor for that neurotransmitter. And once that receptor interacts with the ligand, the neurotransmitter, it's going to cause the whole depolarization, EPSP or IPSP, on the postsynaptic cell. So you basically have just sent a signal across that barrier with a ligand binding a receptor using that neurotransmitter. So this is special because this means you have some control over the type of signal that can get sent, the type of receptor that can get activated. The same neuron can produce multiple types of neurotransmitter, and depending on the situation, that postsynaptic cell can put in the receptor it needs for that particular time and place. So it's a great little mechanism, synaptic transmission. And of course, if you think about pharmaceuticals, what are pharmaceuticals? A good chunk of them are just taking advantage of this ligand receptor binding. And we know there's many of them, especially in the realm of mental health, that are basically pharmaceuticals that either block or encourage the binding of with those receptors on the postsynaptic cell. So these are some of your most common neurotransmitters, and I'm sure you've seen a lot of these, right? Adrenaline, we think of that as fight or flight response, right? Noradrenaline is also involved in that, as well as overall mood. Dopamine is part of your reward pathway. Serotonin is part of mood, right? It calms you down, right? Serotonin reuptake inhibitors, they're often used for anxiety, in that they basically cause that serotonin to not get taken up again by the presynaptic cell and not get metabolized and recycled. Instead, it prolongs the effect of serotonin in the synapse, causing that sort of anxiousness to kind of like the mellowing to happen, right? GABA, so gamma aminobutyric acid, again, these are what they look like, right? Acetylcholine, especially in muscles, is very important. We'll talk about acetylcholine plenty. Glutamate, I already talked about. Endorphins are one of those feel-good molecules that, they're produced in large quantities and many different types. They're different from the rest in that they're produced in a very specific way. They're kind of a pseudo-hormone, but they also have effects on the neurotransmitter, right? So all of these things have particular functions. Depending on the type of cell, the type of neuron, the type of part of the brain, they could be doing different things. So you know what neurotransmitters are. I could talk all day about neurotransmitters, but there's not much to talk about. You know what they are. You notice how they're all kind of similar. They all have that aromatic ring, very interesting, or something like it. So they call them all interact with similar types of receptors. So depending on how much interaction there is, you can alter the course of that connection. So here's some data. This is showing EPSPs and over time. So with stimulation, repeated stimulation, you can reach a point called tetanus. Tetanus is basically sort of a locking of a constant signal. So in this case, you interpret that as the synapse is always doing something. So here is a before and after picture, right? So before, you have a neurotransmitter produced by the axon, you have the postsynaptic density, and then after something called potentiation or long-term potentiation, which just means repeated stimulation, right? What do you see? Well, you see that the presynaptic axon is producing a lot more neurotransmitter, and on the flip side, that connection's gotten a little stronger. It's gotten a little bit physically bigger in this case, but also you see a lot more receptors, which are shown here as those little red things in the membrane there in the postsynaptic cell. So your synaptic density has gotten bigger, and then you, on top of that, have more receptors and more neurotransmitters. So what's going to happen to the signal? How are they connected? Well, you're just going to get more more action potentials, right? Stronger responses, stronger EPSPs between these two, right? You have more neurotransmitter, more receptors, more of them are going to bind. They're going to cause a bigger change in the electrochemical gradient as a result. This is the product of repeated stimulation, potentiation. You know potentiation by another name. You call it learning. And this is essentially the simplest form of learning. Long-term potentiation, or LTP. Long-term potentiation was first described in Aplysia californicus, which is the sea hare, which is a type of nudibranch, which is a type of sea slug. And the advantage here is that they had a system where they had this creature that has basically a very simple reflex. It's the gill siphon withdrawal reflex. So, long story short, if you touch the siphon of a sea hare, which is on one side of the body, it's going to instantly retract the gills on the other side of the body. Now, the best thing about this is that all of this reflex, which is, it doesn't require any kind of like thought, right? You touch the siphon and then it retracts the gills. Well, the more you touch the siphon, the faster it retracts the gills. It's actually a noticeable thing. It gets better at retracting the gills. The best part about it, from a physiology standpoint, is there's only one synapse involved, because a sensory neuron gets activated when you touch the siphon, and then a motor neuron gets activated when it pulls the gills. And there's only one synapse between them. So this made a great opportunity to isolate the effect of a synapse, and that's what they did. And they would do studies where they would test to see how fast the response happens, and they were able to get measurements of conduction velocity, and can also look at changes in neurotransmitters as well as the neurotransmitter signal, right, i.e. receptors, on the other side across one synapse. And what they found is that with repeated stimulation, well, they got more neurotransmitter, they got more receptors, more calcium, and they got a bigger, faster response. And that is the simplest form of memory, the simplest form of learning, right? It remembered that it's being, touched, and it got better at doing something. It learned something. It's like literally muscle memory, right? When we say muscle memory, this is what we're talking about, long-term potentiation. This stuff applies to us too. It applies to probably everything. The more you use it, the less likely you are to lose it. You stop using it, you're going to lose it. That's what this means, right? So you've got to constantly train your mind, train your body, whatever. LTP is what it's all about. It's pretty amazing if you think about it. So we talk about this, and we'll often use the term synaptic plasticity to describe this idea that the synapse is something that is variable through time. So you can have long-term potentiation where you make that signal stronger, or if you don't, you can get long-term depression. If it's an inhibitory signal, you get long-term depression if you constantly induce that inhibitory signal, right? And if you stop using it, you'll get LTD as well, right? It gets low, it gets long-term, it just kind of fades away. So a lot of this has been focused on glutamate receptors. There's two types, NMDA and AMPA. So NMDA is N-methyl diaspartate receptors, all right, as opposed to AMPA, which is amino hydroxymethyl something acid. Anyway, both of these receptors are involved in bringing in sodium. NMDA also does potassium, and they're basically activated by, they have a cofactor. Zinc is the cofactor in both of them, and they're the main ionotropic glutamate receptor. And depending on the tissue, they're the type of thing you would see. Now, one area especially that's gotten a lot of interest in this and synaptic plasticity is the hippocampus. The more you think of something, the more you memorize it. When we talk about things being memorized, we're often talking about them being encoded in a neuron in the hippocampus. Being able to recall it, being able to bring it back, being able to store it in the first place, this is a glutamate-dependent thing that happens in the hippocampus, and the more you stimulate it, the better you're able to remember it. This is the basis of studying, right? So when you study, your hippocampus, which is where you store your memories, your hippocampus is better able to recall something because of synaptic plasticity brought about by LTP. So moving forward, as you want to learn about the nervous system, remember, LTP is what you're aiming for, right? And that the nervous system is basically an integration and coordination. And it evolved for movement first, relies on unidirectional signaling, right? And you have to build circuits. And you can work on that strength of the connections between them based on your stimuli. So the more you train it, the better it's going to work for you. All right, we'll leave it at that, and I'll talk to you later. Bye-bye.